All right, let's get started here. Good afternoon, everyone. Thank you so much for joining us for our second webinar related to the Sustainable Canadian Agriculture Partnership Wheat Cluster. My name is Jocelyn Valestic, and I'm a producer from Broadway, Saskatchewan, and current vice chair of Sask Wheat's board of directors. I am also chair of the board of directors for the Canadian Wheat Research Coalition, also known as CWRC, if you like acronyms. The CWRC is a collaboration between Sask Wheat, Alberta Grains, Manitoba Crop Alliance, aimed at improving the net relative profitability of wheat for Western Canadian producers. The CWRC facilitates a collaborative approach to producer funding of regional and national research projects in variety development and agronomy, including 4 wheat breeding agreements with universities, Agriculture and Agri-Food Canada, and the Canadian National Wheat Cluster under AAFC's Canadian Agriculture Partnership Program and successor programming. That was a mouthful. Additional regional projects that align with variety development and agronomic priorities will also be considered for funding through the CWRC. I will be your moderator for today's webinar, and before we get started, I wanted to quickly run through some housekeeping details. All participants are muted for today's webinar, and if you want to ask a question, please use the Q&A function, which should be available at the bottom of your screen. We'll hold all questions until the end of the webinar and answer them then. Today's webinar will focus on priority 2 of the latest Sustainable Canadian Agriculture Partnership Cluster funding, which is about economic growth and opportunities for wheat. We are pleased to have 2 researchers join us today to talk about their research activities more in depth and how anticipated incomes will support growth opportunities for growers. We would also like to thank the co-funders of these projects, which include the Grain Farmers of Ontario, the Grain Farmers of Quebec, the Atlantic's Grain Council, and CCAN. These partnerships are crucial to plant breeding in Canada. I'd like to introduce our speakers today, starting with Dr. Helen Booker, who is Associate Professor and a Grain Farmers of Ontario wheat breeder, and leads the wheat program at the University of Guelph, which focuses on improvement of winter wheat production in Ontario. Her research program focuses on germplasm, trait development, and improving selection efficiency for breeding of winter wheat. The program has a large field component that includes field-scale experiments at the Elora Research Station. Thank you for joining us today, Helen. Next, I'd like to introduce Dr. Michael McElroy. Dr. Michael McElroy is a research and recognized plant breeder at the CEHRW, the Center of Grain Research of Quebec. He earned his PhD at Dalhousie University in Halifax, Nova Scotia, investigating the interactions of different species of legumes and grasses in pasture production, and continued his postdoctoral studies at the same institute working on cannabis genomics. He started his role as winter wheat breeder at the CEROM in 2017, and since that time has been involved in the development of hard red wheat varieties for Eastern Canada, as well as exploring the potential of genomic techniques for improving selection techniques. Thank you for joining us today, Michael. Both Dr. Booker and Dr. McElroy have prepared a short presentation for their research to date. So Helen, I will let you present first. Take it away.
Okay, thank you. Okay, I have to share, right? Here we go. Can you see the slide, or should I move our faces around here?
Looks good to me.
Okay, thank you. Okay, thank you for the introduction, and thank you for the opportunity to speak today on our project, a cooperative approach to improving Canada Eastern soft red winter wheat to strengthen agriculture sector resilience. Okay, I have to go down here. So why winter wheat? The winter wheat in Ontario rotations increases row crop yields and profits. And these were the results of the long-term rotation studies that were done at the University of Guelph, at the Elora Research Station, and at the Ridgetown campus in Ontario. So this is where wheat is grown in Ontario. So you can see primarily in the southwest and central and in the eastern parts of the province, and the market classes that are grown. And the major market class that is grown is Canada Eastern soft-red winter wheat. And this is the market class that this program focuses on. So our project objectives include development of soft red winter wheat with improved traits, including superior yield and winter hardiness. And this is accomplished by hybridization, followed by selection in segregating populations, and then agronomic testing of our peer lines or elite lines for adaptation to production systems in Ontario and Eastern Canada. Another objective is evaluating wheat for improved resilience through disease resistance to Fusarium head blight, powdery mildew, and wheat rusts. So this is my group at Guelph, from left to right. Mike Pavon, Zhang Hang Zhang. Riley McConaughey is a PhD student in my program. Ropo is a co-op student, and Dr. Mitra Sarah Jazari and Dr. Javid Siddiqui, scientists working in the Guelph wheat program. Co-lead at the Ottawa Research Station, Agriculture and Agri-Food Canada, is Dr. Gavin Humphreys, and this is his group at the Ottawa Station, as well as at the Harrow Research Station. My other co-lead in this project is Dr. Lily Tamburek, and she is located at the Ridgetown campus, University of Guelph, and this is her group there. So one unique aspect of the project is the in-house double haploid breeding that is used at the Ottawa station. So this figure here just shows you the process of double haploid plant production using the maize pollen method. So you can see pollination, application of a growth regulator, and then removal of immature seed containing the haploid embryo. The haploid embryo is then cultured, and the transfer of each haploid plant, you have chromosome doubling using a colchicine and then double haploid production. And to provide maximum genetic diversity among the recovered haploid plants, the embryos of the cross plant or the F1 or the second generation or F2 plants derived from carefully planned crosses would be cultured. So using this technology, Dr. Humphrey's group is able to develop true breeding lines in a single generation. And this technique is widely used in the development of winter wheat cultivars. The other advantage in this technology is merging double haploid production and marker-assisted breeding. So you can have DNA marker analysis and selection of favorable haploid plants that are then further processed with chromosome doubling and double haploid generation. There's also potential here to combine a double haploid production with genomic selection as well as with marker-assisted breeding at the same time. So each of our programs will host an inoculated fusarium nursery. So there is a nursery at the Ridgetown campus, and this is an image here of that nursery. So you can see the mist irrigation. So each of these rows would represent different wheat lines. They would be inoculated with the pathogen that causes fusarium head blight in wheat. And then the mist irrigation would be provided to get the disease to develop sufficiently. So disease incidence and severity can then be scored on each of these different breeding lines. So at Guelph, we're a university-based breeding program. The program is a great platform for graduate student training. This is Riley McConaughey, so he's a PhD student, and he's using machine learning to phenotype for Fusarium head blight in wheat. And he recently released this Wheat Scan R app that counts wheat heads and then isolates them for disease detection from overhead cell phone images of wheat plots. So this figure here is from Riley's first publication based on his master's research in our program. So you can see from the figure, the top left image, a photo of a wheat plot showing Fusarium head blight infection was run through the wheat head detection model. And then you can see in the right-hand side upper image, the segmented wheat heads from the background. And then a spectral index was applied to the segmented heads to separate infected from healthy tissue. That's in the bottom left image. And then in the bottom right image, a histogram was made and a saddle at a threshold of 0.49, it's indicated by the vertical line, was found to separate healthy from infected tissue. So left of the line is healthy and right of the line is infected tissue. So this technology has substantial potential for improving selection efficiency for this hard to improve on trait in wheat. So you might ask, what is collaborative about this project? The three of us, the three public breeding programs in Ontario, every fall we meet to discuss our crossing plans. So we will put forward our parent selections and based on the market class, soft red winter wheat, and the traits that we want to improve. And for the last two crossing cycles, we've been focused on improving resistance to Fusarium head blight in our, agronomically strong, good quality soft red winter wheat backgrounds. So this is just an example of the crossing plan in our program. So the number of F1 plants we'll produce based on the different cross combinations. We'll harvest that seed and that will be planted in the fall and evaluated the following season. We'll also genotype each of the parental lines, and we use CASP markers for this. They are trait-specific markers. And this is what our genotyping looked like of the parental lines that were used in the 2024 crossing block. So you can see the OAC lines. So these are 3 lines that were released from the program and have been commercialized. And some of the lines, CEHRW and other lines from other programs. So here we were trying to integrate some of the disease resistance loci to Fusarium head blight or stripe rust or or leaf rust into our strong agronomic backgrounds. So just a look at the breeding pipeline, which is not unique to our program, but would be used in both at Ottawa and at Ridgetown as well. So we set our breeding targets based on the market class and the trait we want to improve. We select the parents to meet those targets. We tissue sample the parents for genotyping, and then we would make the cross, produce the cross seed. That cross plant would be also grown indoors. We need to produce a number of cross plants for each cross combination to get a large enough F2 population. We would also genotype those cross plants. So for those for those genes that are polymorphic, different between the two parents line, they would be heterozygous in our cross plants, and those would be deemed as true crosses. In our situation, we grow a very large F2 population in the nursery at the Elora Research Station, and this is basically we want to recover those recombinants that are superior to either parent line for the traits that we're trying to improve. And we need a large enough population to do that. We're making selections based on visual characteristics, so plant structure, maturity, height, disease resistance traits, winter survival, all these things we can select for effectively at Elora. Then we'll advance our segregating populations either using a modified bulk pipeline or a single seed descent pipeline. So in the modified bulk, the material gets planted back out at Elora so we can make visual selections again. In the single seed descent pipeline, the material goes into indoors. So at this stage, we're using markers again, markers only at this stage to make selections. So basically, only advancing those lines with the desirable or favorable alleles that we're trying to enrich in our material. And then material goes out either as headrows, planted as headrows, or hill plots, or short rows if it's from our indoor pipeline. And again, we can make field selections based on visual characteristics. So by the F6 generation, the line is more or less fixed for us. So it's a true breeding line by this stage, and it can go into agronomic testing. So we have enough seed to make a replicated test at the Elora Research Station. And it's also at this stage that we start to identify the market class, so soft or hard, red or white. The best lines from our preliminary yield test will be advanced to our advanced yield trial. So this is a multi-location trial. Again, it's replicated. We're making selections based on agronomic traits. We have enough seed at this point to do some kernel quality characteristics. So we have a lot of grain. And based on the market class that the material falls into, it'll either be entered into the orthogonal bread or pastry trial if it's good enough. At the same time, we're evaluating all this material at our inoculated nursery at Elora. As I mentioned, each of us does this. We have registration trials in Ontario are called orthogonal trials. So they're either bread or pastry, depending on the market class. The material is also evaluated at the FHB nursery at Elora and at Ridgetown and at Ottawa. So after 2 years, the candidate lines, the best or the most meritorious, are basically put forward, and they may be supported for registration at this point in time, and then they would be entered into the performance trial. So all the lines that we have in orthogonal trials, we'd start doing our seed development. So we initially grow them as pre-breeder head rows. They're inspected. So the Ontario performance trials, like the registration trials, they're multi-location across all the production zones of Ontario. They're also grown at the Fusarium head blight nurseries in Ontario, and the data is publicly available through the growcrops.ca website. So we also produce our own breeder seed at the Elora Research Station. And also would do our own PBR testing. So at this stage, any lines that are going to be registered, we need to describe them not only for variety registration, but for the PBRO office as well. So the first line that was released from the program, the Guelph program, it's a relatively new program. It was jump-started in 2014, was OAC Constellation. So it was registered in 2020, and it was widely available as certified seed to producers in 2023. And this line is now, or this variety is now, the yield and quality standard for Canada Eastern soft red winter wheat candidate lines in registration trialing. So with that, I'd just like to acknowledge our funding partners. If I can get the slide to move. Okay, it's not really moving. Yeah, it's doing a little bit of a hissy fit here. PowerPoint's not responding, sorry.
Can you list them off on the top of your head?
Yeah, okay. Grain Farmers of Ontario, OMAFA, Ontario Ministry of Agriculture, Food and Agribusiness, Agriculture and Agri-Food Canada, Canadian Wheat Research Coalition, and the Ontario Cereal Crops Committee. Those are off the top of my head. And the last slide was our thank you slide. That's it. Thank you very much. So we almost got through with no technical issues, but not quite. So I'm going to stop sharing.
Okay,
I can get out of here. Yeah.
Thank you so much for your presentation, Helen, and the overview of plant breeding. And it is so complex. So you did a great job of breaking it down. And I know I've seen like even the double haploid technology. I've seen it kind of in person and kind of got the tangible look at the process. And it is very cool and common practice now. So thank you so much for sharing all of that. Next, we will have Michael sharing his research. Take it away, Michael.
Great, thank you very much. So, all right, we can see my screen.
Looks good to me.
Okay, great. All right, well, thanks very much for having me today. I am Michael McElroy, a researcher at the CÉROM, which is the Center for Grain Research in Quebec. For those that might not know us, small but mighty group that's involved in, among other things, wheat breeding here in Quebec. So today we do have a breeding program here that deals with winter wheat. But today I'm going to be talking about another project that's being financed through this group, that is the collaborative approach to genomic selection in winter wheat to accelerate the improvement of complex traits. So, very nice introduction. Thank you, Helen. I can spend less time on this slide right here talking about the breeding cycle. So this is essentially what we have to go through to develop new varieties in any kind of cereal. So about an 8 to 10 year process, going from crossing to multiplication to family selection to line evaluation, and if we're lucky at the very end, we can peel some off and bring that into an actual varietal registration. So we talk about it sometimes as a pipeline, which sort of implies a sort of linear nature to it, but what it really is a loop, because as breeders, what we want to be able to do is get through that process as quickly as we can, so we can put the best material back into our crossing blocks and improve the next generation that's going to come right after that. So that's what we're talking about when we're talking about genetic gain. Breeders are obsessed with genetic gain. And so we're always looking for a way to be able to do that faster. One thing that definitely slows us down is what's on the left side of that circle here, which is the line evaluation. It's very lengthy and expensive to be able to test our lines for all the traits that are interesting to us. And so the faster that we can do that, the better we can do to be able to get the lines that we really want back into our crossing block. And this is where genomics can give us a hand, because really when we're talking about genomics, it is a kind of a look under the hood to see what's going on in terms of the genetic mechanisms that are responsible for all those traits within the wheat. Because technically within every seed of wheat that you have, there is something in the order of 64 gigabytes of information, which is really the map, the road plan for how that wheat is going to grow and what it's going to look like out in our fields. So from a single seed, we should, if we can take a look in there, have everything all the information that we need to be able to select what we want. The real issue that comes from this is being able to take a look at that information and being able to make the link between that and the kind of traits that are interesting to us. So it requires a lot of time, it requires a lot of sophisticated methods, and it takes a lot of data, and that's where collaboration can really come in. So there's lots of ways that we can look in terms of genomics, lots of different methods and whatever, but obviously today we're talking about what's known as genomic selection. So when we're talking about this, what we're really doing is talking about methods of predicting a phenotype of an individual via genomic information. So a phenotype is really just an assembly of all the traits that you would think of when you think of a variety of wheat. So how tall it's going to be, what the end use quality is going to be, what disease is it going to be resistant of, anything like that is really a phenotype. So what we want to be able to do is take a look at the information within that genome of the wheat and be able to say, based on that, can we predict what kind of phenotype it's going to display once we grow it out at large scale. So in order to do this, we start with what we call as a training set. So this is, the same kind of thing when you hear about any kind of artificial intelligence. You start with this training set where you have all the information already available, and you use a model to be able to train on it to be able to predict the things that you want. So in this case, we're using markers that are all the way through the genome to be able to predict these traits that are interesting to us. So it's similar to the kind of stuff that Helen was talking about in terms of marker-assisted selection. But rather than using one specific marker that is linked to one trait and one gene really in the genome, what we're going to do is going to go all across the genome. We're going to pick all sorts of different markers like that all contribute to traits, and use those to be able to create this more sort of complex model to be able to predict these things. So in general, these models are very tailored to kind of a specific germplasm. So, a certain gene pool or however you want to say it of your program for a particular environment, because that's going to change the how they react to different environments. And of course, for breeding objectives, it's going to be different as well. So it's not a one size fits all thing. You really want to be able to create a model for your needs. So to begin to do that, what we need to do is generate a whole lot of markers all across the genome. And this is a very, very basic look at how we can do that. So essentially, we're starting with DNA that we isolate from any number of wheat that we want to take a look at. We cut up the whole genome into little tiny pieces using enzymes. We can then amplify and add little bits onto that to make sure that we can track down which DNA is which. And then we bring it in for sequencing. And essentially, that's just taking a look at the actual genetic code that is written onto that DNA. And so we will sequence all these little tiny pieces, get a whole bunch of little strings of information that we can then compare to a map of the wheat genome as we know it. And we can start to place all these little bits onto the wheat genome and say where they are and how close they are to each other, and start to build a whole picture of the genome of that particular wheat. Once we're done with that, we can start to go through and take a look for tiny little variations that we find in that code, which we call SNPs, or single nucleotide polymorphism. So these are going to be the markers that we're going to use to be able to map out the relationship between these different varieties or lines of wheat. And so these aren't actually going to be the cause of all the differences that we're going to see in the wheat genome, but they're a nice way of figuring out little anchors throughout the genome to spot the differences. Then we're going to go on to the actual model development. So like we said, we're going to start with a training population. So this is a population where we have both genetic and phenotype information. So, we've gotten markers all over the genome for this assemblage of wheat. We've got phenotype information as well, so we know how much each of them yield in a given environment. We know what kind of quality they have. We know what diseases they're resistant to. And from that, we can develop a bunch of genetic models. And with that, we can take what we call the test population, where we only have genotype information, and predict what kind of phenotypes those individuals will have just based on that. And so this can be really helpful in terms of picking out which ones that are really interesting to us. And what's really neat about it afterwards is that we can take that information and put it back into the model and improve it again once we know what the real phenotypes of those selected plants are. Once we can confirm that, we can keep putting it into the model, make it better each time around. So what can we do with this exactly, practically, in a breeding program? Well, there's a number of different things. We can take a look at predicting line performance, which is kind of the classic use of this that we have in plant breeding. So, you know, I've generated a whole bunch of new lines within my program, and before I go out to test them, I'd like to do a pre-selection on them and find out which ones have the best potential to give me the kind of thing that I want. So instead of spending the time and money to go out and test them in the field, I'm going to genotype them all, and I'm going to let the model tell me which ones have the best chance of having the phenotypes that I'm interested in. So that can save a whole lot of time and money in terms of what I put out in the field. There's also a role in that in terms of evaluating novel material. So if somebody sends me some wheat that they think is really interesting, I can genotype it and take a look at what's in there and see if it's interesting to me in the ways that I really want. Are there markers that are associated with traits that are interesting to me? Is it likely to be adapted to my environment or not? Or is it really not worth the effort to try and integrate? We can also go on to simulating crosses. And for anyone who's managed a dairy herd or anything, you'll know that this is kind of what genomic selection was initially developed for, is for really figuring out how do two different individuals mesh together, because we can not only predict existing lines that we have genotypic information about, but we can also invent new ones based on crosses that we have. So if we know what the genotype of mom and dad are, we can put them together, create a bunch of new individuals, hypothetical ones, and say which ones are going to give me the best kind of combination together, which ones are going to mix well. So that can be really important, really powerful use of this technology within a breeding program. So how do we use it exactly within this? Well, to take a step back, and again, I'm repeating myself a little bit with what Helen has already said out there, for applying it to Eastern Canadian winter wheat. So for a little bit of background, we have about 500,000 hectares of winter wheat seeded every year in Eastern Canada. The vast majority of it's in Ontario, but it has started to move really beyond that. There's a lot more going on in Quebec right now. There's a lot more going into the Maritimes. So it's a crop that is definitely expanding. If you take a look at the Eastern Canada in general in the cereal growing region, it's quite a diverse environment and climate. You know, we've got some very humid places, we've got some less so, we've got some quite north for Eastern Canada anyhow, and quite south. And as Helen had mentioned, we've got two major classes of wheat. So we've got the Canadian Eastern hard red winter, which is hard grains and strong gluten, which is used for a number of different kind of breads and noodles. And then we've got our soft red wheat, which is definitely a soft grain, softer grain, low protein. It is for things like cakes and pastries, crackers and whatnot. And that'll come back to us in a moment. We also have 4 different public breeding programs. So again, myself at CÉROM, Gavin Humphreys in Ottawa, and Helen and Lily at University of Guelph at Guelph and Ridgetown, respectively. So generally, the two of us in the north are looking at hard red winter, and those in the University of Guelph are taking a look at soft red winter, but we both dabble in each other's. And we both have, all 4 of us have similar kind of priorities. We're all looking to improve yield and agronomics. We're all trying to improve winter survival. We're looking at getting better resistance to diseases, especially Fusarium head blight. And we all want better end use quality, although our priorities there diverge a little bit in terms of what exactly we're looking for. So it sort of made sense for us to come together and say, how can we really pool our resources to get the most out of this genomic sort of technology? Because like I said, the more data that you have, the more diverse the kind of panel that you're looking at, the more that you can really get out of it. So it really just makes sense. So this was really the crux of the project that we proposed. It was to create a set of genomic tools that would permit our 4 programs to really implement GS easily and more easily trade material. So to create a sort of base for this, what we decided to do was to create a training population of 352 lines, so that's 88 of each of ours, put them together and generate the kind of data that we needed to develop these models. So within that pan, we've got some current varieties, we've got breeding lines, so ones that are in sort of process of being tested within our programs, and some other ones that are parental lines, so ones that we're using as sources of certain traits but might not be interesting in and of themselves. And so this is really to kind of get the 4 corners of everything that exists within Eastern Canadian winter wheat germplasm. And what do we want to feed into this model? Well, I mean, essentially all the stuff that we want to use for our breeding objectives. We want to take a look at agronomic data. So we're testing all this panel in our 4 different sites over 3 different years. We want to take a look at disease data. So we're getting some fusarium data that is going to be coming out of inoculated nurseries, like Dr. Booker had mentioned previously. And we're also going to take a look at some grain quality data, so protein and grain hardness, because these are the things that really separate those 2 classes really well. On the genomic side of it, we managed to generate about 53,000 markers all throughout the genome, which will help us sort of distinguish our different lines from each other and what they have in common and what they have differently. And also genotype the whole thing for 23 single markers, so the kind of things that we'd use for marker-assisted selection. So put it together, this will allow us to develop these kind of models. And what the output that we really want from this is a web-based platform where we can have common models among our 4 programs that we can use for predicting key traits, tools that we can all use to be able to simulate crosses, and a kind of library of genomic data and performance data for all the lines that are in our training set. So we can kind of peruse through this and see which ones might be useful for our programs and which ones we could ask to be able to get some material for crosses or for testing or whatever we want. So it's an updatable thing with new entries being added as they're used. This is really only the first phase that we want to do in this project. So what could we do with genomic selection tools within our programs? Well, within each of them, there's plenty of things that we could do. Just as some examples, we could pick out lines that have potentially yield and fusarium resistance, which would save us a lot of years of testing. We can test early generation material to see, for example, which ones might be lacking the genes that we need for quality parameters, allowing us to sort of reject those right away and then move on to ones that are a little bit more interesting. And we can simulate crosses to figure out which parents would be the best ones to give us, for example, higher yields with less chance of lodging. So some really interesting stuff that we can do within programs. But I think the real strength is what we can do across programs. Because we can, with that sort of library that I was talking about, we can go and take a look at each other's programs very easily and find out what other lines might be able to mix with ours really well and give us the kind of traits that we really need for our own programs. We can take a look at new sources of genetic variation that are missing from our own kind of breeding portfolios and sort of increase the amount of genetic diversity that we have and really create bridges between the 2 different types of programs that we have. We always find it's kind of risky crossing hard red and soft red wheats because it's difficult to sort of pick out the quality that we want out of these. But there's really good points on both of these that I think would benefit everyone if we were able to mix a little bit more. And these genomic tools are the kind of things that will allow us to be able to combine these and still pull out the stuff that we need and keep the traits that we want without losing that end-use quality that we really want. So just to put it very simple, collaboration is the lifeblood of plant breeding, and genomics can really make it more effective. So with that, I'll just say, I got to do my thank you note. So, so I just want to say thank you to our whole team. So Helen, Sylvie Cloutier at ORDC, Gavin, Lily, Frank Yu at ORDC, and Vijendra Kadka, who's working between CÉROM and ORDC. Some help that we've gotten from Tara and Emma for running the markers, our technical staff at all of our programs that help make this happen, and our funding partners that Helen has already mentioned. So I'll just cut myself off there.
Excellent. Thank you so much, Michael. And I could really appreciate that there's the challenges of the different climates, even within a small area, and the models that you get for the winter wheat genomic selection. I also love that you highlighted the different types of wheat that have entirely different end-use products, and all delicious. But both are needed for each market to make sure that farmers can have those markets. So thank you very much. Let's jump right into the questions. First, I'd like to start with Helen. Let's go back to you're talking about what goes into developing a new wheat variety. And can you tell us about how recent variety that's come out of the program and what it's brought to farmers?
Oh, some recent varieties. Okay. Well, we've recently released we've released five varieties since 2020. OAC Constellation, OAC Vega, OAC Moon, OAC Twilight, and OAC Virgo. And 3 of those have been licensed to CCAN. So that's OAC Constellation, OAC Moon, and OAC Virgo. And since the beginning of this SCAP project, we had a line that was supported earlier this year. It was OAC 23 SRW 16. So that all of these varieties were soft red winter wheats. My co-leads have also released varieties as well. So, basically I mentioned the pipeline because there's, products coming out all the time. So it's an evaluation and development process. So yeah, we're constantly releasing and licensing new varieties.
Excellent, thank you. And Michael, let's move to a question for you. How does genomic selection help further achieve research outcomes for farmers?
Well, that's a good question. I mean, essentially for us breeders, it gives us the tools to be able to advance our programs more quickly and more efficiently for the creation of new varieties. So, I mean, the better that we can do our job, the faster and the better the varieties that we can put out and give to farmers, which is a net benefit to everyone.
Excellent. Thank you so much, Michael. We have a couple of questions in the Q&A. We have an anonymous person asking, Helen, it appears significant resources are being put into FHB resistance at Guelph breeding program. Is that the limiting factor for wheat production in Ontario, or is winter kill equally important, and what advances are being made towards winter kill? If you had more funding resources, what bottlenecks would you like to improve on? That's quite a big question, or maybe two separate parts to that, Helen.
Sure. Yeah, thank you for the question. Yeah, so we have focused the last 2 crossing cycles on Fusarium head blight. We had in a previous cluster, we had a Canadian winter wheat adaptation trial. And so we evaluated that material for Fusarium head blight and identified material that was resistant at our nurseries. So we've included some of that material. Now that material is not necessarily the right market class, or it's a feed wheat, AFC Harfang, but it harbors some of those major QTLs, and it was very resistant. So that's why we've done some top crossing, and we're screening fairly large populations. But that is a focus because in Ontario, it's a corn, soybean, wheat rotation, right? And corn is also susceptible to Fusarium. Gramineira infection, so it causes gibberella ear rot in the corn. So it limits the rotation if you have a susceptible wheat as well. And the material that we have released from the program, great agronomics, great quality package, but the weakness has been fusarium resistance, and that's why we focused on that. Winter kill is a hit or miss, right? It happens infrequently, but it does happen on some occasions. We're not. It's harder to identify, a specific line with winter hardiness, but we are, through the project with that Michael's leading, looking at some of the major loci for frost tolerance. So that's how we're approaching that more complex, harder to select for trait because it doesn't happen every year. Like really, we probably get a significant winter kill every 7 years. So it's not something we can select for easily in our nursery, although we're selecting for winter hardiness every year. We can more efficiently select for FHB because we use an inoculated nursery, so we know we're not getting any escapes. Whereas with winter kill, you have to have the right winter to evaluate that trait. Maybe Michael can speak to some of the major genes that we're looking at in our material to determine winter hardiness.
Sure. Yeah, no, I mean, one of the parts of the genomic selection project that we'd mentioned here is taking a look at some of these single markers that have a large effect, and one of them is taking a look at what they call copy number variation in frost tolerant genes. So there's some wheats that just have a lot of copy of the same sort of gene, and that seems to give them an advantage in terms of frost tolerance, at least. So we have some markers that are good for that. I'll say that in Quebec, where winter survival is still very much a problem, though, like Helen says, infrequently. It's definitely more complex than frost, though, which makes it really tough. It's a lot of that winter kill seems to happen really late in the season when we've got those freeze-thaw cycles. And I don't know if it's just frost at that point that's really killing them, or a lack of oxygen, or what, but it's a mini obsession of mine. So I'm hoping to figure out some more about that in the years to come.
Excellent. Thank you, Helen and Michael, both for your answers there. It's definitely, we're going into winter right now, and we're thinking of those little baby plants and how the heck they can survive the winter, but they can. And that's the beauty of winter wheat and the breeding that goes into it as well. So we got another question here. It's a long question with a few different questions in it. So it starts as I presume as winter wheat breeders, you're not able to utilize contra-season nurseries, but are there other strategies you are able to utilize to speed breed winter wheat? Are either of your programs utilizing gene editing tools? How do you see big data and AI impacting your breeding programs? Is any germplasm developed in Western winter wheat breeding programs utilized in your breeding programs? Let's start, Michael. Which you grab one of those questions and run with it.
Well, I'll start with the last one there and say that there is definitely some Western winter wheat that is being used in my program. I'll say especially for the quality, because there's been a lot of really, really great work that's been done in end-use quality in hard wheats in Western winter wheats, and that's something that I'm still trying to advance a bit in my own breeding program. So yeah, I definitely appreciate the work that's been done already, and it's definitely giving some life to mine and helping improve the gluten strength in Canadian Eastern hard red winter.
Excellent. Thanks, Michael. Helen, do you want to tackle gene editing tools or AI, big data?
Well, basically, Riley's project is using machine learning. So basically using imaging to predict disease. And that's going to have huge implications for our selection efficiency for Fusarium head blight and wheat. And it's been hard to improve for trait. We haven't really made any big leaps or bounds in that area. So, this is where we're, phenomics, that's where we're focusing. Gene editing tools. No, we're not using any gene editing tools in our program.
Yet?
Yeah. Yeah, it's if we had all the money in the world, yeah, but it's not practical from financial standpoint for us anyway. Yeah.
Okay.
We'll leave that to the private sector.
All right. So the next question is from Jeff Reed. Helen, can you comment on the Big Six trial, what it is and how it helps accelerate our progress in Eastern Canada?
Oh, okay. So the Big Six trial was a similarly like the group in the Great Lakes region states. They had a similar type of idea to our project on genomic selection in winter wheat. So all the public breeding programs in the Great Lakes region of the states got together, and basically they were doing a similarly to our program, where they were trialing their elite material at locations in the northeastern region of the U.S. The idea was to develop genomic selection tools for those programs. Well, we had this Great Lakes wheat workers meeting, and found out about the program. So I reached out and said, would you like to trial your material here in Ontario? And they said yes. So Dr. Tamburk screened the material at the inoculated fusarium nursery at Ridgetown. We did some agronomic testing at Elora, as well as Gavin Humphrey's group did some agronomic testing of the lines at Harrow. And so we were able to identify material from those public breeding programs in the US that had good adaptation, high yield to our environments, and good FHB resistance. So it's now, we're using them as crossing parents. So that's what we're getting out of the program, and they're getting more agronomic data, disease data for their for training their genomic selection modeling in their programs. So it's really the Western wheat is great, but what our production systems are more similar to what they experience down there. So it's less of a jump for us to use their material. Presumably, that freeze thaw that kills sometimes the winter wheat. The material from northeastern US has a more extensible root system, and so it's better able to tolerate that kind of temperature fluctuations that we'll experience in the winter here than out west. It's mainly like low temperature, whereas here it's a whole bunch of different things that are going on. So either from a winter hardiness background, it's great that we can have access to this material from our US collaborators. Sorry, that was very long.
Great answer. I'm going to ask one more question to end it off here for both of you. How does your research support economic growth for growers, and what additional benefits will wheat farmers see from your research? Michael, you want to take this one first?
Sure. I mean, in terms of developing genomic tools to help us with breeding, any help that breeders can get to create better varieties is going to mean more economic growth, because, you know, if we can improve productivity, if we can cut down on margins, then, you know, that makes for more returns for a farmer, and a more stable and resilient wheat industry in Eastern Canada. And in terms of other benefits, you know, we don't always talk about the sort of environmental benefits going along with economic growth. But, you know, genetic solutions to these kind of things can bring a lot of improvement in that way as well. You know, the less amount of fungicides that you're putting on, the fewer times that you have to go out into the field. If you're harvesting earlier, there's not as much need for energy intensive dry downs or anything like that. So we can address a lot of these environmental problems as well with genetics. And again, we can, the better tools that we have to move this forward, the better it'll be for everyone.
Excellent answer. Helen?
Okay, so we know farmers are paid on volume, so we want to improve yield of winter wheat, and I think that's been you know, happening. It's the OMAFA cereal specialist Joanna Fowlings this past field season, winter wheat broke a yield record, so it reached 100 bushels per acre. So that's a record for winter wheat. And Ontario producers need a competitive small grain. The long-term rotation study showed that winter wheat, you know, improved the resiliency of the cropping system and basically improved the yield and profitability of the whole system. But basically to improve, you know, to maintain that competitive small grain, we need to make some improvements, right? So we need to focus on, well, I'm focused on Fusarium head blight resistance. So it's essential because basically the wheat becomes an inoculum for a corn crop becomes an inoculum for a wheat crop. So if we can, you know, improve that trait alone, I think we've really helped the whole value chain, because, you know, Fusarium damaged kernels are unmarketable. And I think in 2016, it cost the industry $1 billion in Canada. So any improvements in that area, I think, will go a long way.
Excellent. We have run out of time for our discussion today, and a great discussion it was. Thank you for sharing all the information. Thank you to Dr. Helen Booker and Dr. Michael McElroy for sharing your amazing talent and research. And researchers like you both are what keeps plant breeding moving here in Canada. And as a Canadian farmer, I would like to thank you both for providing these field-ready varieties for us to grow in our fields. Thank you, everyone, for attending this today. It is much appreciated that you showed up in your busy day. This webinar is the beginning of more research cluster extension plans. So if you like today's webinar, please stay tuned for more content coming in the next few months. Today's webinar will be recorded and shared on the CWRC website soon. Watch your email and social media for more information about the video when it's live. Thanks again, and have a great day, everyone.
Thank you.
Thank you.